The first step to analyzing proteogenomic data is to filter out events that are debris and doublets. In addition, if multiple samples were pooled using hashtags, we need to demultiplex and assign cells to individual samples. In this video, we will review the workflow starting with count files, the process to filter and demultiplex the data. Start by pointing your browser to mass.cognitive.com. If you do not already have an account, please reach out to BioLegend's technical services. Once you log in, you can create a new dataset either by clicking on the plus sign here or from the side panel by clicking on Create New Dataset. Give the new dataset a name. We should note that a dataset may contain data from multiple experiments or lanes. Within MASS, we refer to a data from a single lane as a file set. Here we will focus on loading data from files in sparse format consisting of barcode, feature, and matrix files that are typically output from Cell Ranger. Loading data from other file formats is also possible. Please reach out to our technical support if you are working with data in other file formats. You can load data by simply clicking on Select Files and pointing to the feature, barcode, and matrix files on your disk. If your multiple samples have been pooled with hashtags and these features are labeled with HTO in their name, MASS will automatically identify these features and show them to you as being selected as hashtags, and will use them later during the demultiplexing process. Depending on your network bandwidth, upload may take a few minutes, but if your network is interrupted during the upload, MASS will auto start from where it left off. Once the upload is completed, MASS will present a summary of hashtag staining for the respective samples. In general, you should expect that the high percentile values for each of the hashtags are indeed high, and the low percentile values are low, because we expect hashtags to have stained specific cells in a positive manner while being negative for the other samples that were not stained with that hashtag. Once you've verified the hashtag staining here, you can click on Begin Filtering to start filtering data by various criteria. The first set of filters are based on UMI counts. UMI counts are plotted with the rank for the UMI on the x-axis and the value for the total UMI for each event on the y-axis. And the UMI plots are created separately for each of the types of features, HTO, ADT, RNA, as well as the sum total across the features. You can click on any of these plots to change the default filtering that is applied by MASS automatically. In general, on a UMI knee plot, you should expect debris to be at the right bottom part of the curve, where the rank is high and the total UMI, given that we are dealing with debris, should be low. On the other hand, on the top of the curve with a low rank should be any doublets that have a very high UMI value and greater than singlets in general. You can change this filter by dragging the box and clicking on Save. The next filter to rule out any cells that are dead, as indicated by high mitochondrial RNA content. You can access the mitochondrial RNA filters by clicking on the mito filter here. In general, MASS will default to a value of 0.8 on the y-axis, which is basically the fraction of mitochondrial RNA UMI as a fraction of the total UMI for each cell. If you want, you can change this filter as well, like in the case of UMI filters, and click Save to apply new threshold values. The next set of filters are based on isotype expression, multiple of which are not expected to be simultaneously positive on a given cell. Therefore, any events with multiple isotypes being simultaneously positive will be excluded. You can set the threshold for how many isotypes are allowed to be positive simultaneously by changing this number, and mass sets this to 2 by default. Like in the other cases, you can click on any of these plots and adjust the gates on each of the isotype. The last set of filters are based on atypical protein expression, that is, pairs of proteins that are not expected to be co-expressed on cells. For example, CD3 and CD19, one a T cell marker and the other B cell marker, we do not expect them to be co-expressing on the same cells. Therefore, very likely these events are doublets and not cellular events. MAS will auto-detect several of these pairs, assuming your feature file contains these protein names, and automatically create these inclusion filters. Like with any other filter, you can click on this plot and adjust the locations of these filters and save it down to create a new filter. At the top of the filtering page, you will see a summary of all the events that were excluded from downstream analysis by the filters, and you can toggle these filters on or off and then save it down in case you want to turn any of these filters off for your analysis. Once the filtering is completed, the next step is to demultiplex, assuming your data set has samples pooled with hashtags. You can do that in the demux tab. MAS will automatically demultiplex based on the HTO count data. These plots here show the positive negative expression for each of the HTOs. And you can adjust the threshold for the positivity for each HTO by clicking on the plot and adjusting the location of this gate. In general, we expect a sample to be positive only for the cells belonging to a sample to be positive only for one HTO and none of the other HTOs. The summary table here shows the expression for each HTO and each of the samples for the sample that it is associated with, and the expression of that HTO in the other samples that it is not associated with. As expected, you expect a higher count for this sample and low counts for the other samples. And the last column here shows how many cells were attached to that sample based on that HTO expression. You can also click on one of these rows to visualize which events were identified with that sample and the expression of that corresponding HTO on those cells. Once the demultiplexing is completed, you can proceed to the summary page, to examine the staining across all the samples for individual ADTs and HTO. The first few columns in this table here provide statistics for the counts for each ADT for singlet cellular events. In general, we are expecting an antibody to have stained at least some cells. Therefore, we are expecting a high value for the 100 percentile or even 75 percentile. In addition, unless there are some experimental reasons to expect otherwise, we expect the staining pattern to be similar for an antibody across the samples. The level of expression for a given antibody can easily be verified from the first few columns. The later columns, one per sample, can be used to understand the uniformity of staining across the samples. The values shown here indicate if there is higher or lower staining in a given sample compared to the rest. The values are 0.5 minus the Mann-Whitney U, which measures the overlap between two distributions. The values close to 0 indicate no bias, while a value closer to 0.5 would indicate a strong higher staining for a sample compared to the rest of the values, while a value close to -5 would indicate low staining for a sample compared to the rest. You can search for a given any desired marker and examine how the staining pattern looks. For example, here, CD3, you can click on that row and examine the staining for all events. for the events that were predicted as singlets across all the samples, and finally the staining pattern in each of the samples. And as these numbers indicate, in general, CD3 staining seems to be similar across all the samples, but perhaps with a small bias, lower staining bias for the middle sample here, which you can actually see in the curve here, where you can see this positive peak for CD3 in this sample slightly offset compared to the other peaks. You can also visualize the expression of CD3 across all the samples, the 3 clusters from demultiplexing, and you should expect reasonably similar staining across all of the samples. At this point, the filtering and demultiplexing steps are completed, and you are ready to proceed to the next step of identifying highly variable genes and normalizing the RNA, which is presented in the next video.